upon us. This Brain to Society series is one of three webinar series, the one where we bring the science of real-world behavior together. And I am extremely pleased today to have Susan Carnell, a close collaborator and a colleague who has been trained originally in psychology and progressively getting layers of neuroscience and of public health. And what she will be presenting us today, it's a series of work on the understanding through primarily neuroimaging study, but how does the human brain in kids and adults, but our focus is on adolescents, how are those decisions of food being made, and how can there be intervention, innovation to make this adaptive behavior kind of the default option? A bit more on Susan's vitae. She received her PhD from University College London, postdoc at Columbia. She is currently associate professor in the Department of Psychiatry and Behavioral Science at Johns Hopkins. A research program, as I said today, the focus will be on neuroimaging, but she used a full portfolio of methods, including behavioral tests, questionnaires, genotyping, hormonal assay, and so on, to really tap into the multi-scale influences on eating and obesity in children and adults. Before letting Susan present her seminar, just a bit of additional information of the overall program of the hour and a half, 2 hours we will be spending together. Our centers have, over the years, pompously, I can say, advanced this convergence science, which is to say, how can we build, bridge, and interface between those various scales of influence? So Suzanne's presentation will be followed by a very rich panel that each of them intersect with some angle of her work. One of them is, for instance, studying the same type of decision making in adolescence in rats. Others will be looking at the social capital context and so on. So we are talking about 45 minutes of Suzanne's presentation, during which everybody will be muted, including myself. And then after that, we will be having a first round of panel where each panelist is invited to follow up in some way on what Suzanne will have presented, and or add one layer to our understanding of obesity risk in children. We will also then open the floor for everyone. And at that time, I think we unmute everybody, or if you have questions, you put them in the chat in the chat window. I think that's all. So welcome, Suzanne, and I'm very much looking forward to your presentation. Suzanne, the floor is yours.
All right. Well, thank you very much, Laurette. I'm very happy to be talking to you today in these unusual circumstances. Due to the COVID situation, there's not quite as much new data as I would like in this, but there is some. So even those who've heard me do my thing before, hopefully there'll be some new information in here. So as Laurette mentioned, I'm going to focus today on brain imaging studies of appetite in children. As we all know, we live in what's been called an obesogenic food environment. This is just a picture from the cafeteria at Johns Hopkins, which has been doing its best to kind of improve the offerings. But as you can see, there are still a lot of palatable, very high calorie, energy dense food cues tempting us to eat. But what's interesting to me and to many others on this call are that there are still significant individual differences in body weight. And so I've been interested in thinking about why that might be. This is true, in adults, and it's also true in children, which is my particular interest. And so on a voyage of trying to find out why this is, as Lorette has already alluded to, I started my journey in the UK, where I'm from. And really my first work was in child eating behavior, thinking about how we measure that, how we conceptualize that, and working on a concept of called appetitive traits, which I'll explain in a second. And then my early work really was focusing on the influence of parent feeding on child eating behavior and weight. And then I became very interested in the influences that were coming from the child. So that's when I got interested in genetic influences and started doing some twin studies to explore those. I then started thinking more about the actual genes, and I was studying this during a time when FTO emerged from the GWAS studies as the first common genetic variant to be associated with obesity. So I began becoming interested in that. Then I moved to New York, and there I really started following this biological direction a little bit more, and did some work in gut hormones, and started working using fMRI, which is what I'll be focusing on today. And then my journey so far has culminated in Baltimore, where I'm really trying to bring all these influences together. So I think this fits quite nicely with Lorette's goals, which is why we've been collaborating for a while now. Everything that I do is with a range of collaborators at Hopkins. Is everyone just able to see my slides here, or do you see kind of other windows? Does it?
No, it's very well, it's very good, Suzanne. You can go ahead.
Great, I'm just kind of moving some things around. So I'm lucky to work with some very talented young people in my lab, and I want to kind of acknowledge them up front. Many of them are on the call today. Elena, Geeta and Kimberly Smith are postdocs. Jenny Sadler hopefully will be starting with us soon. I'm also going to talk a lot about work which has been on fMRI, which has been analysed by Liu Chen. And Anna Hirs is a research coordinator in our lab, and a lot of the work has been also contributed to by Aphrodite Papantoni and Leora Benson, who are now in other labs but still working with us, and we collaborate with these senior collaborators that you see on the slide there. So first I want to present what I'm going to call a biobehavioral susceptibility model, which describes how I see all these different strands of my work fitting together. So what this proposes is that children differ in things called appetitive characteristics. So one example of this might be food cue responsiveness. How much does seeing all these high palatable food cues in the environment make us want to eat those foods? And then what I propose is that interacts with our levels of appetitive characteristics interact with the obesogenic food environment to drive our energy intake and ultimately our adipose levels. Okay, that works better. I've been particularly interested in genetic and home environment influences on these appetitive characteristics. And then what I'm really trying to do now is bring the brain into the picture and ask what MRI particularly might be able to tell us about how genes and environment are affecting our appetitive characteristics. What might be the neuromechanisms for those phenomena? So I'm going to start by actually describing some behavioral studies, because this is where kind of all my interest began, and it motivates all the imaging studies. And I want to talk a bit more about this term I've used several times already, appetitive characteristics. So what do I mean by that? What I mean is enduring dispositions towards food, or you might think of them as eating styles, that differ between individuals. So an example might be food cue responsiveness, as I mentioned earlier, and another might be satiety responsiveness. So how responsive are you to internal cues which actually tell us to stop eating, which might be things like the release of hormones in the gut. Are these things associated with weight? There does seem to be good evidence that they are, from both behavioral tests and from questionnaire measures, and I'm just going to show you a few examples here. So one test that we and others have used quite a lot in the field is called the eating in the absence of hunger test. In this particular example, we administered it to children in their homes, and we also had a separate sample of children in schools. And we gave them a lunch, which they ate to satiety, and then we gave them access to a bag of palatable snack foods. Here, a bag of Cadbury's chocolate snacks, which anybody who's tasted Cadbury's will know they're pretty palatable. While they had access to this bag of snacks, though, they also had access to some alternative activities, which in this case was a puzzle book. And what we did was to measure their intake in the absence of hunger, and to relate this to weight. And what you can see in this particular study is that there's a relationship with weight such that those who were in the overweight and obese groups were eating more in that test. And you can see that the relationship here is much stronger for boys, which I think could be to do with various factors, which we can kind of discuss. But I think it does speak to the importance of using various different methods to assess these appetitive traits, because it could be that there are kind of situational influences which are making girls and boys behave differently here. Although this is just one test I'm showing you, this was from a study in the UK, there's also lots of other evidence for cross-sectional associations between eating in the absence of hunger and weight and adiposity in children. This has been done in children ranging from 3 to 18 years old. And there are also some evidence for longitudinal associations. So we see that eating in the absence of hunger earlier in development seems to predict weight later in development. So the eating in the absence of hunger test is generally thought to tap into that food approach or food cue responsiveness aspect of appetite. Another test which has been used quite a lot in the field is the caloric compensation test, and I'll describe that to you a little bit further. So this is thought more to tap into that satiety responsiveness trait. So for this particular test, what we did here was to administer 2 preloading tests. So in each of these tests, children consumed a drink preload followed by a standardized lunch 30 minutes later, and that happened on 2 separate days. However, on one day, the preload was relatively high in energy, and then in one, the preload was relatively low in energy, and these conditions were counterbalanced. We actually did this in 2 different forms, so we had different types of preloads. On the first test, we had a high compared with a low-calorie orange drink, which actually tasted identical. So the high-calorie version was made by adding maltodextrin. So these were kind of organoleptically indistinguishable, but they had different amounts of calories. For test 2, the different preloads were water versus milkshake. And the important thing about this paradigm is that we can calculate the degree of compensation for the preload calories. So we can calculate an index called COMPEX, which is reflective of the degree to which children adjust their lunch intake based on the caloric content of the previous preload. And what you can see here is if we just look at mean compensation across both of those tests, is that it's associated with weight groups. So the children who are in the overweight obese category actually compensated less well for the preload calories. So some indication from that test that higher or lower satiety responsiveness seems to be associated with weight. And this has been demonstrated in other studies too, in children ranging from 4 to 17 years old. Ours were 4 to 5 years old, using a range of different preloads and meals. But as I mentioned before, these tests are very informative, but they can be influenced by situational factors. And obviously they're not practical for assessment of appetite if you want to do large scale kind of research or intervention work. So what we've used instead to be able to kind of scale up our research in that way is something called the Child Eating Behaviour Questionnaire, which is a parent report measure. And I'll just go through that a little bit now, because I'm going to refer back to it a few times in the talk. So this questionnaire measures a number of traits which we might think of as being associated as described as food approach. So one example is food responsiveness. So you can see the items here. An example might be, my child is always asking for food. We have a scale measuring enjoyment of food. My child enjoys eating. Desire to drink. My child is always asking for a drink. And emotional overeating. My child eats more when anxious, for example. In contrast, we also have scales which try to assess food avoidant traits. So we have scales measuring satiety, responsiveness, and slowness in eating. For example, my child gets full easily. Fussiness, my child enjoys tasting new foods, reverse scored. And emotional undereating. So items like my child eats less when he or she is upset. Quite early in my career, we conducted some behavioural validation of this scale and found that indeed those scales do associate well with behavioural tests of appetite, like caloric compensation and eating in the absence of hunger. We showed here that they do change through development. So what you can see here is scale scores at 4 and at age 11 in the same children. And you can see that in general, the food avoidant traits are going down over that age span, and food approach is going up. In that context, though, there is tracking of these traits through childhood. So from that same study, you can see that there were quite strong correlations between values at 4 and values at 11. So there is some tracking of these traits too. And they do seem to be associated with adiposity. So this is just one example from a large study we did in the UK, where we saw that children with higher waist measurements had higher reports of enjoyment of food on that questionnaire, and also lower satiety responsiveness. There have now been a number of studies done using this questionnaire in many different countries. It's been translated into lots of different languages and it's freely available if anyone's interested in using it. But a recent systematic review and meta-analysis we did just kind of demonstrated that we do see strong associations, positive associations with adiposity for the majority of the food approach traits and negative ones for the food avoidance traits. Less data is still available using longitudinal designs though, so that's still important to do in the future. What about babies? So we know from epidemiology evidence and preclinical work that really obesity risk begins in infancy. So we wanted to be able to see whether we could measure these traits in infancy. And to do that, we've used something called the Baby Eating Behavior Questionnaire. I won't go through this in detail, but you can see that we have traits which correspond with the CBQ measures, specifically enjoyment of food, food responsiveness, let's move this window, slowness in eating, and satiety responsiveness. And these do seem to be associated with infant growth. So this was quite a nice study that used a sibling design. So it actually looked at same-sex dizygotic twin pairs who were discordant for BBQ scores and looked at growth trajectories. And what this is showing here is that there was more rapid growth over time from kind of 0 to 15 months in the siblings who had the higher food responsiveness. And conversely, there was also more rapid growth in siblings who had lower satiety responsiveness. So that's quite nice design because it controls for genetic impacts and also some home environment impacts to show that BBQ scales do seem to be predicting infant growth. So to just summarize from that research, it does seem that food responsiveness and satiety responsiveness are measurable in infancy and do predict more rapid growth into toddlerhood. And so I've been interested in trying to explore where these characteristics come from. And as I mentioned earlier, a great way to do this is to use a twin study. So we can start to look at the relative influence of genetic as opposed to environmental influences. And this is data from the TED study, the Twins Early Development Study. This was a large sample of 8 to 11-year-olds. And what we were able to do was to look at the correlations within monozygotic as opposed to dizygotic twins in satiety responsiveness and enjoyment of food, and to use that to generate heritability estimates. So what you can see if you just look at the graph here is that correlations are obviously much higher within share all of their genes. And if we calculate the heritability estimates, we can see that about 75% of the variation in enjoyment of food and 63% of the variation in satiety responsiveness is genetically influenced. Interestingly, when we look at the other appetitive traits, we see a slightly different picture. So food fussiness also seems to be heritable to the same degree, but emotional overeating and emotional undereating seem to show more environmental influence. So some variation in the relative contribution of genes and environment within the CBQ. What about appetite in babies? This is showing you the same kind of graph, but for infants. And again, you can see that these bars are all higher than these bars, which indicates greater similarity in the monozygotic twins for each of these scales. And if you look at the heritability estimates, you can see that they're highest for those food avoidant traits, slowness in eating and satiety responsiveness, and substantial but more moderate for enjoyment of food and food responsiveness. Which genes are implicated? Well, as I mentioned, when the FTO gene variant was discovered in 2007, we genotyped our large twin sample. What we found was that children who had the obese genotype on FTO had lower satiety responsiveness. And since that time, there have obviously been lots of other behavioral studies which do seem to suggest that this FTO variant and actually polygenic obesity risk estimated by summing the contribution of genetic variants which have been identified in subsequent GWAS, they do both seem to influence appetite and eating behavior and in turn body weight. So some indication about which genes we might be tapping into for some of our twin study results there. So now I'm going to move into what is the main focus of the talk and start talking a bit more about neuroimaging studies, which have been designed to kind of build on those results. So I'm going to take you back to my diagram here. I'm going to talk about two aspects here. So I'm going to talk about first the influence of familial obesity risk on neural responses to food cues. And to talk about familial obesity risk, I'm going to use the model of maternal obesity. And the reason that I'm interested in that, of course, is because if we use a familial risk model, we might be able to identify neural risk factors, which could be predictors or mechanisms of future weight gain, which are actually unconfounded by current obesity. So parent obesity gives us this nice model to look at genetic and environmental influences on our outcomes of interest. So what I did here was to look at neural responses to food words, actually, in adolescents who varied in familial obesity risk. So the subjects were post-pubertal 14 to 18-year-olds, 10 of whom were obese or overweight, and 26 of whom were lean, of whom 16 had an obese or overweight mother, and 10 had a lean mother, biological mothers. And they underwent 2 visits, an initial consultation and then a test day in which they had a pre-fast meal of 2 bottles of Boost at 12 p.m. They had a blood draw, that was so we could look at FTO and some other SNPs. And then they underwent an fMRI scan followed by an ad libitum multi-item meal. Just checking the time here. Okay. So I'll speed up. So this is the food cue paradigm. You can see we showed pictures of food words. We had high energy dense foods, low energy dense foods, and we also had a non-food control. And you can see they were all designed to look like a menu on a restaurant chalkboard. And then after each of these stimuli, they had to rate their appetite on this scale. I want to eat it, ranging from not at all to extremely. And then we also had a control rating for the non-foods. And this shows you the results for the for the wanting scores, for the lean low-risk adolescents, the lean high-risk adolescents, and the obese overweight adolescents. And what you can see is if you look at this column, we're seeing higher wanting scores from the high-risk adolescents. We were expecting, though, to see similar values or higher values for the obese and overweight adolescents, and we did not. I think there's a number of reasons that could have explained that, and we can kind of talk about those later. One possibility could be social desirability. Maybe they didn't want to kind of eat as much in that setting, or express their wanting for the food in that setting. However, when we look at the ad libitum meal intake, we do see more of the predicted relationship there, with intake kind of increasing with degree of obesity risk in more of a sort of dose response manner. No effect, though, with fruits and vegetables, which you can see we had very low intake across all of the groups there. So some indication for increased food responses at a behavioral level among the high risk group that could perhaps promote excess weight in the future. But what about the brain data? So this was primarily a neuroimaging study. What do we see here? I'm showing you a lot of brains here, but I'm going to kind of talk you through it and highlight what I think some of the most important findings are. So this shows you results from our food compared with non-food comparison. And what you're seeing in the middle is data from the lean low risk group, the lean high risk group, and the overweight group. And then what you can see at the sides is the results from the contrast of the overweight compared with the lean low risk group and the lean high risk compared with the lean low risk group. And one of the first things to point out is that across all the groups, we're seeing this deactivation of the default mode network, which I think makes sense because they're kind of engaged in this salient task, and food is salient to everybody. So we're seeing that across the groups. What you're also seeing is that the lean groups seem to be showing increased activation of areas implicated in self-regulation. So that's quite clear, even if you just look, look across the images. You can see you're seeing a lot more red for the lean groups in that kind of self-regulation circuit, which is very similar to the regions that you see activated in tasks involving response inhibition, such as the go-no-go task. What you're also seeing, though, is that the overweight group is distinguishing itself a little bit here. So the overweight group is actually showing deactivation of the infralateral and dorsolateral prefrontal cortex here. This shows you results from the high compared with low energy dense comparison. And here you'll see that we're seeing a lot more red in the lean low risk group as compared to the other groups. And so for this contrast, we really seem to be seeing that the lean low risk group is showing that increased activation of the self-regulation circuit. The lean high risk group here seems to be showing a deactivation of the dorsolateral prefrontal cortex. So to kind of summarize that a little bit, it does seem to be that familial obesity risk in adolescents here was associated with relatively lesser activation of a self-regulation circuit during food evaluation. Now, this was just a cross-sectional study, so we unfortunately can't tell whether this predicted their weight, but this does seem to be something that could relate to weight gain in the future. So now I want to move on to talking about results from a new study that we're just kind of analyzing now. And for this analysis, I wanted to focus on the neural correlates of appetite using a similar paradigm. So we recruited mother-adolescent dyads for a further study of familial obesity risk. They again went an initial visit where they had height and weight and waist and body fat measured, and also mothers completed the CBQ. There were 2 visits. They arrived at the lab around 3:30, and then they had a fixed liquid meal or preload at around that time. But we had 2 conditions. We had a fed condition, where that preload was a 480 cal drink of Boost, or they had water, which was a fasted condition, and that order was counterbalanced across individuals. They then underwent an MRI scan, which included a food cue task, and then they had a multi-item ad libitum dinner meal. So this paradigm enabled us to get several assessments of appetitive characteristics, the psychometric measure of the CBQ that I mentioned, and then we also were able to extract a number of behavioral measures of appetite, and I'm going to focus now on that measure of ad libitum intake, I'm going to focus on data from the fasted condition, and also caloric compensation. So the degree to which individuals compensated for the calorie they consumed in the fed condition by eating relatively less at their subsequent ad libitum meal. Here, we actually switched from food words to food pictures. So food words had the advantage of being very easily controlled across the different stimulus conditions. And then also people would just imagine their favorite example of an ice cream sundae. But we also wanted to try giving people a more kind of salient cue, which might increase food motivation a little bit more. So for this paradigm, we presented high energy dense foods, low energy dense foods, and non-foods again. This time we gave them a wanting to eat rating using a 4-button response. And you can see the trials that one run contained 45 trials, which were randomized, and we had 2 runs. And so we were able to generate a food compared with non-food contrast, and also a high energy dense compared with low energy dense food contrast. And here is the picture of the multi-item ad libitum meal, which was kind of loved and loathed by my lab because there would always be lots of leftovers. So people are probably a little bit relieved that we stopped data collection for that particular study. These are the descriptive characteristics. Because I'm running a little over, I won't go through those, but I can talk about those in more detail if people are interested. This shows you the association between CBQ food responsiveness and BMI-Z score. So you can see we saw a positive association, and also with waist circumference. So we kind of replicated that well-known finding. And what I'm going to show you now is areas which showed greater activation to high energy dense compared with low energy dense food cues with higher CBQ food responsiveness scores. And these are pictured here. So you can see we saw greater activation in the insula and also in the putamen. And interestingly, we also saw an overlap of neural profiles when we did this same, we conducted this same regression for enjoyment of food scales, emotional overeating scales, and scores, and the ad libitum meal intake. So just to kind of highlight that these patterns of activation you're seeing, they kind of differ in strength, with the food responsiveness being the strongest pattern of findings here, but they are kind of similar across these different scores. However, for the emotional overeating result, we also see greater activation of the cerebellum. So I think this is pretty interesting. This is kind of pretty early work, of course, but I think it does suggest the promise of using neuroimaging to examine shared and perhaps unique biological bases of these different dimensions of appetite. So what about satiety responsiveness? I've been talking about food cue responsiveness so far. Well, here we were able to, as I said, generate an estimate of compensation by using that fed versus fasted design. So just to tell you the behavioral effects of this here, what this is showing is ad libitum total meal intake in the fasted and fed conditions. So what you can see is that intake was lower in that fed condition. Fullness was higher in the fed condition, and desire to eat was lower in the fed condition. And if you look at the individual data points, what you can see is that, this is a modest effect, but there's a definite kind of relationship there, which we see, and there's substantial variation in between individuals. So that gives us something to associate with our fMRI data. When we look at the relationship between caloric compensation and weight, you can see it's not overwhelming. So there does seem to be a suggestion of something there. Definitely the much heavier children seem to be compensating a bit less well, but that's not a significant relationship in this data. Oops. Excuse me. So how does this relate to the brain data? So what I predicted here was that better compensation would be associated with greater activation of self-regulation regions in the fed compared with the fasted condition. So is that what we saw? Well, what I'm showing you now is areas which show greater activation to the food compared with the non-food cues in the fed compared with the fasted condition with higher compensation. And you can see that these regions here emerge. So better satiety sensitivity as assessed by that complex score was associated with greater food-related activation of these, of the thalamus and the supramarginal gyrus, which have been associated with sensory integration. Notably, these are not those more cognitive self-regulation regions, which we saw in the earlier study. So I'm looking forward to kind of discussing all those results with you at the end. I just wanted to present some hot off the press results, which Geeta, our new postdoc, has been working on. So when Geeta arrived, she came with a background in fMRI and also analysis of structural MRI. So she was interested in looking at whether a familial risk might also be associated with measures of brain structure. So as those on the call will know, the cerebral cortex is a highly folded sheet of grey matter that lies inside the CSF and surrounds a core of white matter. And cortical surface area and cortical thickness, which can both be assessed via MRI, are independent measures, with grey matter volume being a function of these two indices. And higher cortical thickness seems to be particularly interesting, such that higher cortical thickness is associated with cortical development and has actually been associated with higher IQ in later development. So Geeta wanted, in our study, to look at how familial obesity risk is influencing grey matter volume, white matter volume, and also cortical thickness. And these are the results which she's generated. So I'm going to talk you through what you're looking at here, and then I want to talk about one specific finding which I find very interesting. So what you're seeing here is areas where there were kind of different differences in grey matter volume, white matter volume, and cortical thickness, or CT, in the lean high risk compared with the lean low risk group. So these are the grey matter results, the white, these are areas where we saw lesser volume in the high risk group. These are areas where we saw greater volume in the high risk group. These are the white matter volume results. So these are areas where we saw lesser volume in the high risk group. These are areas where we saw greater volume in the high risk group. And these are the cortical thickness findings. And what I want to do is just highlight this finding I think is particularly interesting in relation to my last study. So we saw that the lean high risk group was showing lesser grey matter volume in the anterior cingulate, and also lesser cortical thickness. And if you kind of compare that to our previous results, you can see it's actually in a similar region. So this shows you the previous results where we saw lesser activation to food cues in that region in the high risk group. And in this study, we're seeing reduced grey matter volume and cortical thickness in that same region. So I think this is, whoops, sorry, I keep having to move this window which pops up. I'm sure I'll have to get rid of it. I probably can minimize it. So if you put those results together, I think there's some suggestion that familial obesity risk may be associated with both decreased volume and cortical thickness and decreased responses to food cues in the food cue task in this area of the anterior cingulate. So we're particularly interested in exploring findings in that region going forward. All the work that I've mentioned so far has been in adolescents. I'm also now trying to extend my work into earlier age groups, because we know that appetite and obesity risk start being formed much earlier in life. So I'm now working on something called the Resonance Cohort, which is actually funded by ECHO, which is a large program across the US, which is funded by the NIH to examine environmental influences on child health outcomes. So the Resonance Cohort, which is the one I'm collaborating on, is a composite actually of a number of different sub-studies which were initiated within the last few years, some of them beginning while the mother is pregnant and some beginning in early childhood. It's an accelerated longitudinal design, and the currently combined cohort now has well over 800 children. Actually, these numbers are a bit out of date. We have anthropometric data, dietary recalls, biospecimens, sociodemographics, cognitive and behavioral tasks and questionnaires. And we also have a wealth of neuroimaging data, both structural and functional, to map fetal and postnatal brain development. So we don't have any task data, but we do have resting state data in these kids. And this just shows you some preliminary results associating maternal obesity as assessed pre-pregnancy, and resting state connectivity. So this just shows you data from 214 data sets from 88 children. So 24 of those children had obese mothers, 19 had overweight mothers, and 36 had normal weight mothers. And this was a selected sample which was matched for factors like gestational age, birth weight, family, socioeconomic status, family size, parity, infant feeding in terms of breastfeeding, and gender and ethnicity. And what you can see here is that children of obese mothers so far seem to be showing increased intra-hemisphere and decreased inter-hemisphere connectivity. So just some very early results there and a taster of the kind of things we might be able to look at as this cohort progresses. We are also going to be extending the measures that we assess in this cohort with a focus on child appetite. So we have a sub-study, which we're currently calling Nom Nom, although my postdoc Elena pointed out we may have copyright issues there, so I probably need to think about that a little bit more. But the goal of this study is to enhance measures of appetite and obesogenic risk factors in that whole resonance sample, and also a sub-cohort who are selected for high compared with low genetic obesity risk. And so what we'll be able to do here is test relationships between early brain development and development of appetitive characteristics, as well as with obesity relevant genetic and environmental factors and child weight trajectories. So once the COVID restrictions have lifted and people are a little bit happier coming back into the lab, we hope to be able to administer an ad libitum buffet meal and also an eating in the absence of hunger test, and also to explore some possibly explore some new measures. So for example, tests of delay of food gratification. This is my partner Jason kind of modeling what that looks like to some degree. The reinforcing value of food. We're also thinking about introducing a measure of taste reactivity. We've kind of discussed this with Patricia. We'll see whether that might be plausible. And we also, of course, have CBQ data mounting up and BBQ data, which we'll be able to look at. So I will now summarize what this, the data I've presented show, I think, so far, is that individuals do seem to differ in appetitive characteristics. These characteristics are measurable in childhood and infancy and are cross-sectionally and prospectively associated with weight. And they do develop and change over time, but they also persist over time. So we see tracking in individuals. Weight-associated appetitive characteristics seem to show a substantial genetic influence. And tests of neural food cue responses are revealing that familial obesity risk is associated with lesser activation of a self-regulation circuit, and food approach assessed directly with the child eating behavior questionnaire seems to be associated with greater activation of areas subserving food reward. And maternal obesity seems to be associated with indices of slower brain development in early life. And I showed you some functional data there, but we also have some structural data which suggests that. So lots of potential future directions, and I'm sure we'll get into some of those in the discussion. I'll just throw a few out which I'm interested in. So I think even before we get to the imaging part, there's a lot that we need to be doing behaviorally. So there are still not very many behavioral measures of appetite that work well in children under 3 years. So I think actually there's a lot of scope to develop measures so we can assess that a little better. What I've not kind of explored as much so far are the relationships between these appetitive characteristics and diet. So might it be that your appetitive characteristics affect your diet, and actually vice versa? Could your diet affect your appetite? What does healthy appetite development look like? There still haven't been that many longitudinal studies in this area, so we're not sure yet. So I think there's a need to develop kind of norms and growth curves associated with healthy weight, and cutoffs which might be associated with excessive growth. We could also do comparisons with disorders of low appetite to start getting a feel for what these measures, these kind of numbers that we generate from the CBQ actually mean. A big question is how mutable are these appetitive and appetite related obesity endophenotypes? So, for example, we know now from work in infants that responsive feeding interventions seem to be obesity protective to some degree. So it would be interesting to see whether these are really acting by affecting our appetitive characteristics. Dietary interventions might also affect appetite. And we've actually done some work, which I haven't had time to go into now, which is in adults in bariatric surgery. This seems to affect one's taste responses, and we've shown that kind of behaviorally and using neuroimaging. Kimberly Smith has been leading that work. So perhaps there is potential to intervene to actually affect people's appetitive characteristics. In terms of neuroimaging, I think we need to know what are the neural correlates of kind of persistent appetitive and appetite related obesity endophenotype. So there's still not a lot of work on reliability of these kind of neural responses to food cues. Do people who show heightened responses at one time also show heightened responses at another time? I think we kind of need to establish that and know what are the neural correlates of these kind of the persistent neural correlates that we can see. Also, what are the neural correlates of development of appetite, of appetitive and appetite related characteristics in early life? That's something we hope to be able to look at in the resonance study. So I focused here on direct measures of appetite, but we're also interested in more general measures of self-regulation and how they might relate to food self-regulation. Are the neural correlates of these shared or are they distinct? We'll be able to look at some of that, I hope, in resonance. And lastly, what are the genetic and environmental influences on appetitive characteristics assessed at this kind of neural level? That's also something I hope to be able to get into and have begun exploring a little bit with Lorette's group. So I just want to acknowledge these funding sources. I've already acknowledged my lab, and now I just want to acknowledge my children. These are now outdated pictures, but they definitely inspire my hypotheses about appetite and brain development. Here's Helena eating an apple in a proud mommy moment, but you know, this was taken a while ago, and now she's nearly 4, and her favorite food is still goldfish crackers. So even as, you know, child eating behavior specialists are not, you know, not able to get children to eat a purely healthy diet. So I'll finish. I know that we'll have a panel discussion now, so I'm excited to kind of hear the panel's discussion and your questions. And I'm also going to just give a quick plug. We're doing a appetite survey, interested in seeing how people's diets have changed during COVID. So maybe I could share that with you guys afterwards. We'd be interested in survey responses to that too. And I'll finish there.
Thank you very much, Susan. I always love not only your enthusiasm intellectually, but also the comprehensiveness of the multi-scale knowledge you have of all of this. And before we move to the panel, I'd like to take my privilege as a chair to push a bit the thinking of this brain to society toward really moving what insights, what solution can we propose? And there was one of your most recent, your slides at the end, where it's quite unusual to have both the structural and the functional fMRI. You had this results on family obesity risk that were related to the decrease in volume and cortical thickness, as well as in the responsiveness in the ACC, the anterior cingulate. So from what you know of the brain, from what you know of what drives behavior, how should we be thinking of moving from such science to say, okay, where should we be starting to find solutions?
That's a big one. Hit me with the big one.
I know. Okay.
Yeah. So you're saying, like, what are the consequences, basically, of these imaging findings? Yeah, I mean, I think that's so important. I mean, what I get from it is that, I mean, or certainly what kind of arose for me from that first study is that the differences we were seeing were really in adolescence, at least, about this kind of self-regulation aspect. So maybe or that was kind of the circuit that was highlighted. I mean, that was obviously because of my population, because of the task design, but it seemed that it was maybe less about, at that stage, about the kind of appetitive kind of the food cue responses, and more about how we make the food decisions and how we deal with that. One way, there is a strand of work which tries to kind of strengthen people's self-regulation. I know there's a group in England that's doing that, and they actually do seem to see generalization from kind of cognitive tasks where you're just trying to prevent a response. They actually do seem to see that helps people inhibit their responses to food in life and affect their weight. So that's kind of one possibility. I definitely think those kind of behavioral interventions could be relevant, but I'm sure we'll also be discussing, environmental level interventions that could also be relevant.
Yeah, in exploring this cascading of insights between the brain and the actual impact, we will inaugurate our panel by Anita. And sorry, Anita, if I pronounce your name wrong, Anita Touche. She is an assistant professor at Queen's University in psychology and economics, having trained in Germany, then postdoc at Caltech and at Max Planck's Institute. So Anita, why don't you keep elaborating what Suzanne is suggesting from the perspective of getting really to real translational brain to society, and any other comments or question you may have for Suzanne in about 3 minutes or so.
Thanks so much for the introduction, Lorette. So let me first say, Susan, I really enjoyed your talk. I love this kind of multi-layer, multiple kinds of data combination approach to address these kind of questions. But one of the things that I was really curious about, and which I think is really closely related to what Lorette just mentioned, is whenever we do these kind of neuroimaging experiments, we always have to ask ourselves, what do we learn? Yeah, what might the costs of acquiring these kind of data actually worth collecting? And so one of the things that really spoke to me during your talk was the question, what does these kind of group differences in the between high risk and low risk people actually mean? With a specific eye on these kind of brain areas that have been suggested to be involved in self-regulation, cognitive control. And so one of the questions that I had was, what is the mechanistic understanding? Because I believe that having this kind of mechanistic understanding will allow us to do what Lorette just asked, to develop this kind of novel targeted interventions, helping us to address some of the problems that people have in the way that how they make decisions. Do you know what I'm saying? And so I was actually wondering if you have any thoughts, what exactly the DLPFC or the DACC do, and what drives this kind of group differences that you have observed. And so you touched on that a little bit, saying that it might be this kind of inhibitory control, but honestly, I'm not so sure that that's actually it. So because I feel like it's a very simple story, it's very easy to sell, but I'm not sure if it's the full story. And so here's where I come from, and I would love to have your take on that. So there have been a couple of studies using the following framework, that a lot of these kind of decisions that we make in daily life, they require us to integrate sometimes competing considerations, such as, for instance, health and taste of food items, right? And so the way that we integrate them is by assigning some weights to these kind of different features that we need to integrate. And so it has been suggested that areas such as the DLPFC might actually be involved in up and down regulating the weights that we assign to these kind of different contributing factors. And so I was just wondering, and again, if this is the case, then issues such as the saliency of food cues or stuff like that might be the way to go to actually help people adopt a healthier diet. So this is the kind of framework where I come from. And I was just wondering if your neuroimaging data speak to that issue.
Yeah, well, I love that comment. I think it's so important. It's making me think that, so I do think a lot of the reason that I saw that kind of circuit activated was because I wasn't just showing people food pictures. Like, people have done previous tasks where they just do a block design, they're showing high calorie foods and low calorie foods, but I was actually getting a response. And actually, for that first study, not for the second study using food pictures, but for the first study, we asked them to rate how much they wanted to eat it and then how much they felt they shouldn't eat it. And that was because at that time I'd kind of read the work looking at food cues, and I'd wondered, well, what does this frontal cortex activation mean, and is it sort of people are trying to do this cognitive task? So I think so it kind of was a food decision task. Like, it's a very basic food decision task, and I've done that deliberately because I want to start working with younger kids. That's why I haven't quite gone down the kind of neuroeconomic route, which I did, could think about a little bit. So I wanted to keep it super simple, but it is a task where they have to rate their wanting to eat food, and then in this first task they're rating how much they want to eat it and how much they feel like they shouldn't eat it, which kind of is like a health sort of related decision. So I actually think it was a food decision task, and so that's what I was making kind of people do. So I guess that's how I think my data might speak to that. But I mean, I'm thrilled to establish this connection and talk to you about the actual kind of mechanisms. I'm not sure. We do have a collaborator here, Vikram Chib. I don't know if you're aware.
Sure.
In neuroeconomics. So, we're hoping to kind of collaborate a little bit more on that end. I mean, I'm kind of interested in the ACC obviously is a very diverse region, and it does kind of lots of different things. And I know that some of the findings from neuroeconomics, what they're calling the ACC, is almost in kind of motor type areas. So I wonder if is that the case?
It depends. It depends what literature you look at. So, I mean, honestly, when I looked at the DACC, one of the things that I was also thinking about, which came to mind, is there is evidence showing that brain structure in the DACC, and pretty much the same kind of area that you showed on your slide, has been involved in individual difference, or be correlated with individual differences in your ability to delay gratification in a monetary domain, as measured in these kind of intertemporal choice tasks. I don't know, so for people that are not familiar with this kind of area of research, the basic idea is that you ask people to decide between smaller amounts of money now or a larger amount of money later on. And so the idea is that if you're better at delayed gratification, you will wait to get more money out of it. And again, these kind of structural differences in the DACC have been correlated with individual differences in the ability to do so. And so that's actually another question that I had. If you have any idea whether these kind of neurocorrelates that you identify also extend to other domains, where you might need to resist the temptations of some immediate rewards. Because I feel like dietary choice is obviously one of the things that we are facing on a daily basis. But the general idea and the mechanism that I was alluding to earlier on might generalize to a lot of different domains that require the same kind of mechanism. So I was wondering if you have any data speaking to that.
Yes, I love this question. So, I mean, we did try and assess alcohol intake, substance use a little bit, and I'll say it was just kind of very, very low in our sample. So I don't think we've got any, that was kind of what I was thinking in terms of real life. We have a measure of food-related delay discounting and money-related delay discounting.
Oh, really?
Yes, we do. And I wanted to get that together in time for this talk, but I haven't managed to. But we will be able to look at that for sure. I think we have run some association with the risk groups, and I don't think they were straightforward. We also have some socioeconomic variability in our sample, and from what I recall, I think that there were some differences which were kind of related to that, with the lower SES individuals showing kind of rate of delayed discounting. So I think we kind of need to unpack that a bit more. But we do have kind of the food versus non-food version. We also have a food go, no go task, which we did in the scanner. And so I think we will be able to get a little bit about, I think that speaks a little bit to your question. We did keep it a very simple go, no go food go, no go task. So we basically had a high calorie food and a low calorie food, and then they kind of made the responses to those. So there weren't lots of different categories within that. So we kind of kept it very simple. And that was because we wanted to take the cognitive load out of it, because we were kind of wanting to extend this to younger kids. However, I think that means that the results we're getting might actually be just more like a normal go, no go test. So yeah, my thinking there is that we might be tapping into kind of a general type of inhibition, but that may transfer to foods. I think they're going to be highly related. I think it's a very interesting question. And then obviously, yeah, I mean, I'm struck now that I'm working more with people in cognition, that a lot of the tests we use, say for example, delay of gratification. I mean, people, the original test with that used marshmallows, because it's just this great example of how people do things. So I think there's going to be a lot of overlap. And thinking apart,
but I guess, I mean, you are kind of restricted by the kind of subject samples. I mean, the reason why they use marshmallows was because it was a test, run in children, and they usually don't care as much about money, at least when they are small, as about marshmallows. So
yeah, another point to mention on this is we've done also a study at 8 to 13-year-olds, which I didn't talk about there, and there we do have a delay task. It's actually developed by Kerry Roche. She works with Stuart McCloskey, and that's the reward there is game time. So there's actually a real reward.
Nice.
Whereas what I have in my adolescence is all hypothetical, so needs to be taken with a grain of salt. But that should be interesting. We haven't analysed that yet.
Okay, thank you very much.
Thank you.
We move now to Cecilia Flores, who also studied brain development in adolescents, but in rats. Cecilia is professor of psychiatry and associate member of neurology and neurosurgery at McGill University. And she is also, she studies rats, but she's also very, very concerned and insightful with all these translational aspects of having impact in real world. Cecilia, why don't you comment as you find appropriate? Next will be Patricia. Go ahead, Cecilia.
Yes, thank you, Susan. Very interesting talk and a very interesting approach and very comprehensive. I really like it. So I have lots of comments, but throughout your talk, I kept on thinking about the dopaminergic system. And when you mentioned the DICC and your findings, I found them very intriguing. So one thing that I was thinking is that group of lean, high risk, I find it particularly interesting. And I was wondering whether you have some information about them in addition of the behavioral responses toward food. If if they have like other phenotypes related to psychopathology, more prone to depression or addiction, anything like that. Because of course, impulsivity and you're using the go, no go task. Reduced cortical thickness would be associated with other phenotypes that probably.
Yes, we do. I mean, we do. When you started talking, I was thinking, well, we don't have anything. We didn't really assess kind of depression or anxiety or anything kind of in the psychiatry direction, but we do have impulsivity. If we weren't broadcasting, I would quickly pull up the results now to check, but I probably don't have time to do the Windows juggle. But we do have multiple measures, actually, questionnaire measures of impulsivity, which we could look at, and we do have perceived stress as well. I can't recall what the differences are there. The lean high risk group are lower in SES, so I think we will see a lot.
I see. Yes,
with that.
I was going to also ask you about that.
Yeah.
I see. That's very interesting.
Yeah, I'm very interested in this, actually, the effects of the socioeconomic environment on neural responses to food cues and eating behavior. This is quite a small study, so we kind of have variability kind of in both, but not really enough to do a really good test of that. But it's something I'm very interested in. So I think we have all the measures to start looking at some of these things, and then maybe it's the next grant that will, or the resonance sample when they age up, will enable us to look at some of those things.
Yeah, because I was thinking that maybe they are like have an increased risk in general.
Yes. And I'm sure that's the case. Yeah.
Including for the studies that you mentioned at the end for the mothers, maybe also like that confers an increased risk in general, and by looking at the imaging and so on, you started to try to find out the neurobiological basis of that, which
Right.
Which probably has to do with the different trajectories
Right.
of the development of the cortex, because it's really
Right.
developing around that time.
Yeah, I think that's so interesting, because you know, we always try and like pick apart like one particular effect, but in reality all of these things go together. Like, you know, maternal obesity does go with SES, and you know, lower SES does have all of these effects. So so if it's only when in, you know, human work, I think when we do those really big studies, we'll be able to tease apart the independent effects. But then I think, you know, what is the meaning of that when things actually kind of cluster together? For example, I know that in our resonance data, we can see effects of maternal obesity, but if you look at SES, the effects are kind of much bigger. So we may be kind of, you know, finding smaller effects that we're interested in, but they're in this kind of broader context, which is having a much bigger impact.
Yeah, I mean, the way I see it is that it would make sense, perhaps, because I'm seeing it from the mechanisms that are happening in the developing brain around that time, that I don't think would be specific for a particular behavior, but for a more general risk, as I said.
Yes, I think that's really true. And then, you know, the question is like how we look at that, because that's definitely the sense, I'll tell you, because we collected a lot of measures in this study. So now I'm kind of looking at everything and thinking, what's the best way of describing it? And it is almost like we see kind of clusters of behaviors. So maybe we do need to start thinking about that more, but it's tricky with a smaller sample, but I think that's definitely what's going on.
Yeah, it's difficult for a smaller. So you're still following them up, this adolescent sample?
We're not. It was not. This was an ROO study, the second part of a K99 ROO.
Oh,
So
I see.
we have their details, and we've sort of tried, but we're not doing that great. But obviously, we have the longitudinal infant imaging study. We're also starting a new study in 8 to 13-year-olds, and that will have a year follow-up within it. So we'll be able to look at some of the similar things in a new sample, but I'm not following up the current one.
Okay. Yeah, because it's very difficult.
Yes.
A quick question to you too. Seeing Tim Mohan's picture in the beginning of your slides, maybe remember first time that Tim and I met at an NIH meeting. Tim is working on motivated choice, completely in rats. We are working on motivated choice in human. So my question to both of you, how do we move? There is a lot of translation that has already been done from years of work on the rats, but how can we move one step further in moving from pre-symbolic to animals such as rat to us human being? So what are your thoughts there for where our translational work can go for the next level of what we can reach? Maybe starting with you, Cecilia, and then Suzanne.
Very, very good question. I will give you some examples that I'm thinking in terms of my own work. For example, we identify a gene that is very important for the development of the cortex in adolescents, and we identify all these in mice. But turns out that this gene is also mutated in humans. It's called the DCC gene. So I collaborated with someone in my department, with Marco Layton, and we actually studied these humans that have the same mutation that we have studied in mice, and we look at the dopaminergic system and about behaviors that we have done in rodents. So now we can know that's a way of translating, right? By using the same genetic changes in mice and trying to identify them in humans. From that, then I started collaborating with Patricia, maybe she can say more about it. And we started looking at this gene and building her very nice genetic risk score using this DCC gene, and we find very interesting results that I will let her describe, but also that we can learn about prediction of who is at more risk, for example, of impulsivity or more comorbidities between addiction and other disorders, etc. In the same way, we've been looking at disorders that are involved in the prefrontal cortex, and we find that this gene is also very important in depression using postmortem approaches. So I think in my case, it's been to use our findings in rodents to guide questions to address in humans. And then we really address them having a lot more information. And the last thing, so as not to take over from this panel, is that we started measuring correlates of the function of these genes by measuring markers in blood.
Okay.
And we can then, our approach now is to try to use this as biomarkers of prediction. And we are now translating our work by some studies in mice, trying to measure these markers in adolescence, and then see later on if there are more at risk for depression in this case.
Very good. That's really insightful in spelling out this multi-scale mechanism that I really believe we need to get serious about. Suzanne, a quick comment before we move to Patricia?
I mean, that's all just so kind of exciting to us. I mean, I think that you know, the interface between, you know, the humans and the animals is kind of so important. I mean, even going back to, I mean, the eating in the absence of hunger test that we do in children, that was based on animal research. The reinforcing value of food test came from animal research. So we've been stealing kind of ideas from you guys for a long time. And yeah, I think it's a back and forth. We need to keep doing it.
Yeah, that's right. And we can keep on being in touch in the future.
Thank you, Cecilia. Shifting to Patricia, who is a very long time Brain to Society collaborator, even if she's still very young. Patricia is a primary investigator at the McGill Centre for Neuroinformatics and Mental Health, and a work building upon the gene brain and life course exposure is also, or life course experience is also one of the key characteristics of her work. And one facet that we have started talking about that I would like, Patricia, in your commentary, as you find if you find appropriate, is that one of the things that I say quite often is that we need to get as serious in assessing the multi-scale mechanism that I was mentioning earlier, but finding a way to assess multi-scale environmental exposure. How do exposure to layers of environment or layers of experience over a life course cumulate the same way that the SNPs cumulate into a GWAS? So Patricia, why don't you go ahead? Daiva would be the next if she's back online. Go ahead, Patricia.
Thank you, Laurette, and thank you, Suzanne, for a very nice talk and for the discussion. It's really interesting to be here. Yeah, I'm glad you brought the layers to the table, Laurette, because my thinking was really going into this direction. But we've been talking a lot about the environment, but I wanted to discuss a little bit about the layers that we have, even thinking about the individual. So you mentioned about differences in brain structure and function and responsivity to the cues. And you also mentioned, Suzanne, about individual differences. So we know that people will differ in this outcome, but this is coupled with behavioral differences, probably in relation to food choices and food intake. But all this is inserted in an individual that has also metabolic differences in terms of energy expenditure, insulin sensitivity, hormonal secretion. So even if we stay limited to the individual, we're not talking about the environment yet, you have like a range of individual differences that happen at many levels, right? And I think this touches a little bit of what Anita said and Cecilia said, because all of that will be deeply associated with the mechanisms that explain the findings that you have. Now, you compared groups of lean, high risk, low risk, and obese individuals, but the groups are kind of the result of this layers, the interaction of these layers. So my question to you and the discussion would be like, what about, what if you don't impose the groups and let the data tell you how do they match together? Did you try, like, I don't know, forming clusters? Did you investigate these differences in terms of metabolism, like insulin resistance, or do you have data on that?
Oh, so many interesting questions rolled into that. I mean, so I will say, so we kind of took this familial risk design, but that basically amounted to kind of, because there were no gaps in the distribution, we basically were taking kind of all comers until the point where we started turning down the obese, overweight adolescents, just because of where we were kind of sampling from. So I do think that justifies taking these approaches where I do like look at all the samples together. So that's what I did for the CBQ. I just assumed that I recruited based on variability in these things. And so now I kind of can look at the sample together with the, acknowledging that it was not, how the sample was recruited. So that's kind of one piece of it. I'm forgetting now what the other pieces were.
Do you have like more metabolic?
Metabolic, yes, good question. So we don't in the adolescents, and we did not assess, because we were kind of at the adolescent stage. For the resonance sample, we're obviously collecting a lot of information about the pregnancy, and so we can look at, say, gestational weight gain and whether the mother had gestational diabetes or something like that. I'm trying to think if we actually, I'm sure we must have assessed whether anybody had diabetes, but we may have excluded that. I mean, these were basically healthy adolescents. Antonio Convite at NYU has obviously done work in adolescents, and he's very interested in how kind of insulin resistance, even like pre-diabetes in adolescents, might be impacting brain structure and function, and he has found some suggestions that it is. So it could be that, even though the obese and overweight kids in my sample didn't have diabetes, maybe they did have some kind of indicators of insulin resistance, which would be driving some of this, but unfortunately I don't have that information.
Yeah, because especially in kids, adolescents, and more in young kids, not necessarily you're gonna have the definite phenotype. Like, not necessarily you need to work with diabetes or overweight or obesity, but the individual differences are gonna be there already, right? In terms of insulin sensitivity and all the precursors before you reach the point of any clinical disturbance. Maybe these features can tell you much more about the biological processes and the groups that form, right? Because this is, as we use functional genomics to try to tell us about biology, but also metabolic features can help. And then you have the many layers like the genomics, metabolomics, brain imaging, all inside the individual, right?
I'm curious as your thoughts on what would be the kind of best measures, because obviously there's always this balance between, so ideally we would like everyone to come in and then we give them, we give them a standard meal and we assess, we put a line in and, stuff that I've done in adults, but then that's kind of harder to do in kids. So we always end up making this compromise. So this kind of new study that we have starting, we're going to get a fasting blood measure, so that would be good, but we're not doing kind of postprandial. But what are your thoughts on kind of measurement? I mean, what can we get at this point that would be informative from just like a pinprick or a saliva sample? What are your thoughts on that?
I think that measures like glucose, insulin, leptin, body composition are very interesting to tell you about which groups are formed. Like, so instead of imposing groups, you just ask the data to tell you how do they group together, right?
Do you think even a fasting kind of glucose from just a one-off fasting glucose would be helpful?
I think it will. Of course, if you're only looking at glucose, you need to be more careful in terms of the conditions in which you collect the sample. The more information you have, the better. But But yeah, even saliva, you can measure a bunch of stuff in saliva.
Yeah, we have in the adolescents. I'm just thinking about what we can do in the adolescents, because luckily we're going to have all of this stuff in the the big study, the longitudinal. But yeah, the adolescents, we took saliva samples for DNA, so I haven't kind of mentioned those analyses, but yeah, we didn't do too much in the way of metabolism, but this is all good future directions.
Yes.
Anything else, Patricia?
No, I think that's
No?
it.
Okay, Daiva.
Yes, I'm here. I am on the phone, so I'm glad there was a backup connection because somehow I lost internet, so I apologize for that. But thank you so much, Susan, for the talk.
Daiva,
So
sorry, I did not introduce you, poor girl.
Oh, that's okay.
Daiva is from the School of Human Nutrition at McGill, having training in nutrition genomics, also very, very active in the Brain to Society. Go ahead, Daiva.
Thank you. Yes, so I actually have a different question from what I was initially going to ask because I was sort of inspired by seeing your So I think Patricia's comments on the individual metabolic differences are very relevant, but it also made me think about individual behavioral differences and sort of neurological behavioral conditions in children. I wonder if your child's eating behavior questionnaire, does it correlate with behavioral conditions in children like sensory processing disorder, ADHD, possibly autism spectrum disorders? Have you looked into that, or have others evaluated those relationships?
You know, we have. I mean, we've got measures of you know, impulsivity, kind of questionnaire measures, and we do see associations with the child's eating behavior questionnaire. I'm trying to kind of recall what they are. I may be able to. I may be able to pull them up later, but we can look at that. In terms of the question of ADHD, I actually am working with somebody who works in ADHD, and when I got to Hopkins, I asked her to weigh and measure all their kids. So we now have quite a lot of structural and kind of resting MRI data in a sample that, you know, varies with ADHD and obesity. And I'll say one of the, we're just kind of starting to look at that data now, and one of the striking findings is that we do see a similarity in I think it's prefrontal cortex reduction, which we see in kind of ADHD and obesity, but then there are also some findings which are unique to ADHD and unique to obesity. So that's just kind of, that there are definitely kind of behavioral correlations between the, you know, impulsivity, at least part of ADHD and eating behavior. And there do also seem to be some kind of neural kind of a shared neural basis and a different neural basis, but so we're looking at that, but that's probably all I can offer so far.
Yeah, no, it's very interesting because I'm very interested in this topic of the gut-brain axis, and science had an issue recently on what is now termed as the psychobiome. So how the microbiome is impacting neurological, psychological factors. I believe that issue is focused mostly on adults, but it would be very relevant to look at childhood and adolescence as well. So I wonder maybe if a future direction could also be to collect fecal samples to look at microbiome relationships between the appetitive traits, the outcomes that you're observing. Any thoughts on the microbiome?
Yeah, I mean that's
aspect?
a whole world that I don't understand well, but we are collecting microbiome in the resonance sample and collaborating with a colleague at Wellesley, that's kind of her expertise. I'm trying to think what's been done so far in that, and I'm not sure. I know that they have done a preliminary analysis kind of relating fecal microbiome to brain development, and they have findings have emerged, but I don't remember them in detail, I'm afraid.
Thank you.
That's okay. No, it's very interesting.
Daiva, anything else?
Well, if I have a chance just to ask about my initial kind of theme was going to be on the issue of taste, because that's where some of my work is active in and looking at individual differences in taste and other sensory perceptions due to genetic variation. TAS1R2 gene and bitter taste sensitivity is a classic example. Adam Dronowski has some nice work on that. People can be very sensitive to bitter taste compounds in foods, particularly vegetables, and that leads to less desire to consume them, less enjoyment, lower food preferences of certain types of vegetables. Have you considered, I know you mentioned something with taste reactivity, but also factoring in some more assessments of sensory perceptions and how that may influence the eating behaviors?
Another great question. One of our postdocs, Kimberly Smith, has a background in neuroscience of taste, so I hope that we'll be able to pursue some of those questions together in the future. We did genome-wide, we used the mega array, so we have the data from a genome-wide array. So we could potentially look at taste variants of interest. I don't know if you have kind of particular ones in mind. In terms of sort of sensory aspects, we don't really have any data, so we didn't get people's kind of ratings of tastance or anything like that in the adolescents. But we have the ad libitum food intake data, so we have kind of some estimates of how they made food choices in that setting, and we also have 24-hour dietary recall data, which we've just for one day, so it's kind of not the gold standard, but we would be able to kind of use that data to some degree, but I don't think we have exactly what you're talking about.
Thank you very much.
Thank you,
Daiva. We move now to Robert Levitant, our psychiatrist on the team. Robert is at the University of Toronto, CAMH, professor of Department in Psychiatry, and I've been interested for long in dopamine mechanism, the RD4, within the context of eating and mood disorder. So Robert, why don't you comment as you found appropriate or follow up with questions for Susan?
Sure. Wonderful talk. It's a talk that I think all our residents in psychiatry should watch, because it was just a tremendous historical review, but also pointing to the future. I am a psychiatrist, and I'm going to approach this from a sort of psychiatric point of view. And I'm going to start off with the whole challenge that we have to study complex common disorders. And the two big ones that I've been interested in my career include obesity and depression. And in both cases, we've seen increases in these disorders over time. So there's something about the environment that must be causing that. And they're also difficult to treat. We find that a lot of times our treatments aren't great, or they don't last as long as we'd like. And one potential solution to that is to define distinct subgroups that are different. Because one explanation for a lack of a treatment response is that you're considering everyone as being the same, when in fact they might be quite different, and in fact even opposite in some ways. And there's lots of evidence for that. So I'm going to sort of talk about subgrouping, because one of the themes that emerged from this talk that was very exciting, I found, was the notion that one form of obesity, in my opinion, really does relate to how you started the talk, which is looking more at appetite and reward and satiety mechanisms. And my best guess is that that kind of obesity is the one that's more purely genetic, probably harder to treat in some ways, sometimes caused by rare mutants, and often a cause of severe obesity as opposed to sort of more moderate obesity. And I think that the FTO genes, for example, are a good set of genes to look at for that kind of sort of almost pure metabolic obesity. But the imaging findings were very striking to me, because they really tell us that a whole other type of obesity relates more to how the brain is actually organized. Because two of the key findings that you reported are that the high-risk groups have problems with brain areas that relate to self-regulation. And another one was that the finding that the two sides of the brains were not communicating with each other as well in a high-risk group compared to a low-risk group. So in some ways, I think both of those findings are showing the same thing. There's a suboptimal organization of the brain in terms of its ability to regulate itself, in terms of different parts of the brain to talk to each other. And I think that this reflects a whole different phenotype of obesity that is much more non-specific. It's not necessarily even related just to eating behavior. It's related more to as you say, you know, general self-regulation and behaviors that cut across not just obesity, but multiple psychiatric disorders, for example. And we see lots and lots of obesity in our psychiatric patients. And the implication, I think, is really quite important, because if there are these two fundamental different types of obesity, I think we really need to study them separately, or at least always think about them separately if we're going to recruit large groups of patients with obesity, because the mechanisms that are, you know, fueling the disorder are obviously quite different if this model is accurate. The other thing about it is that it implies also that a lot of the prevention of obesity doesn't necessarily relate to food. It can relate to self-regulation and helping families that have this vulnerability, starting at a very simple level with low socioeconomic status and general stress and general lack of support in these families. I've been interested in the whole notion of attachment behavior, maternal-infant interactions, and maternal sensitivity, which are potentially amenable to intervention and would be an important way of improving self-regulation and so forth. It also reminds me that, you know, one of the things about modern society is that family structure is completely different now than it was 30 or 40 years ago. You know, we don't have grandparents around, and I'm thinking a lot of the solution to these problems would just be having grandparents around to help support mothers, to help regulate things and regulate families, and you know, routines is something I remember my grandparents were always focused on. Simple things like sleep routines, activity routines, all the non-specific things that are probably most important in terms of improving self-regulation and in terms of brain development and having different parts of the brain being able to talk to each other. It all has to do with probably really simple things that somehow in modern society we've gotten away from just because of the way things have evolved. So I think it's really interesting that a lot of the findings aren't necessarily related just to eating, but rather to self-regulation, brain organization that I would say can cut across, you know, multiple disorders, including depression, for example. Very, very similar sorts of things happen in depression. Also, what's important about that is that from a genetic point of view, if we talk about the DRD4 gene, the good news is that we've done a lot of work with Laurette and we had Belsky last year talk about this. The whole notion of differential susceptibility, and Patricia's paper looking at socioeconomic status in the DRD4 gene. The good news is that if we intervene in these non-specific ways in susceptible individuals, individuals who have what we think might be susceptibility genes would actually have a better than average outcome. So children, for example, with the 7 repeat allele in a low socioeconomic environment might have a better than average outcome if we can intervene in these non-specific ways to help them self-regulate and so forth. Perhaps I'll end it there and see what others might think about that somewhat simplistic differentiation of two critical phenotypes, one very simple metabolic related, the other much more complex, probably the one responsible for the increase in obesity, and much more related to general brain mechanisms and organization.
Thank you, Robert. Suzanne?
I think this is such a fascinating idea. Yeah, I feel like a lot of people are talking about how this is clustering, you know, like we have to think about, you know, comorbidities, not necessarily kind of comorbidities, that's not the word, but there's clustering kind of at the risk factor end, and then at the behavior end, and at the biology end, and increasingly with kind of large samples and, you know, big data approaches, we might be able to get to some of that. I mean, what you're saying makes a lot of sense, to me. You know, another thought is that, like you say, if you kind of intervene, you're going to have, even if you're kind of focused on one thing. Like, I think one of the reasons that these tasks on food, you know, practicing kind of regulation around food might be helping is because they're improving kind of general self-regulation, even if it isn't actually kind of specific to the food. So I think a lot of the effects that we're seeing, probably do kind of speak to that model. Yeah, I don't know if kind of others have thoughts. I think it's a great way of looking at it. I know people have started trying to see if there are kind of subtypes like within obesity treatment. So Kerry Boutel does work in child obesity treatment, and she's tried to see if there are these different kind of profiles of appetitive traits that would actually help her to kind of funnel people into a particular intervention. So for example, maybe someone would benefit more from food training to response prevention around food cue responsiveness, or kind of mindful eating to kind of ride out the urges. So I think people are starting to think in that direction, but that doesn't really take into account the kind of wider forces that you're talking about, which is almost more kind of clustering about kind of general behaviors and environmental conditions. So I agree that sort of factoring all of those things, and then maybe also the metabolic factors would kind of produce the best results in some ways. The kind of counterpoint to that, and something I struggle with when I try and kind of think of the implications of my work, is that you do end up thinking, well, maybe the best intervention is something that's quite simple that actually everybody would benefit from. So for example, your point about into the grandparent intervention, I often think that I often get to the point where I kind of go through all this work, and then I think, well, the best intervention would probably be even not a grandparent, just to send someone round to kind of help a new mom.
Exactly.
Having been there twice. So your points are well taken, I think. And I think a lot of our work, we can start to look at some of these factors, and then I think that has to be in the discussion, because I think this gets onto something different, but maybe something you guys are also interested in. I think the media may leap on kind of one small finding, but actually it's this kind of broader context which is important. And even if we do think that these individual traits are very important, everybody's going to do better if we make an environmental level intervention, and therefore, in an individual who is at high risk of overeating and exposure to these high palatable food cues, if there are kind of less of those around, then they're going to be protected, and it's probably also going to be better for the people who, are lean. So those are my thoughts.
Thank you very much. And that leads very well to our last panelist, that is Spencer Moore, Associate Professor in Health Behavior and Health Education at University of South Carolina, who has been involved with the Brain to Society since its beginning almost, and who has been studying a lot of the social factors that you two are talking about. So Spencer, why don't you get in as you find appropriate? We have 15 minutes to go. We will have Spencer comments and Suzanne's reaction, and then if there are questions from the floor, we will unmute everyone, and you will just have to identify yourself. Spencer? Spencer, you need to unmute yourself. Or can someone unmute Spencer?
There we go. Is that better?
Yeah.
Okay, great.
Yeah, better.
So yeah, first of all, thank you, Susan. I really enjoyed the talk. It's slightly outside of my wheelhouse. So, but I think some of the questions that have been raised certainly fit into the work that I do, and I think one of those is in the terms of these biosocial pathways. I know you're focusing on biobehavioral pathways, but in the work I do around biosocial pathways and approaches, certainly chronic stress and social stressors are a key, mechanism and process by which to understand how social environments really get up under the skin or affect the brain and other types of health conditions and outcomes. And so the stress, and you mentioned yourself having some measures of perceived stress, but I'm curious about more chronic stress, whether we think about that in terms of food insecurity for families, whether it's low socioeconomic status. I guess to pressure a little bit on how you think those types of chronic stressors and sometimes the maladaptive coping behaviors that may arise from those chronic stressors might moderate the relationships between the behaviors you're seeing and the outcomes that you're examining. And just to follow up that, I know you've also highlighted some of the challenges really in being able to study that, whether it's the sample size, whether it's having variability in socioeconomic status of families, perhaps in your studies. So I'm just wondering if you had some other thoughts about how you might get around some of those challenges and really be able to link together, you know, the role of the social environment in influencing family risk, and then looking within families to see how that plays out in terms of children's outcomes. I'll stop there.
Yeah, great points. Yeah, I mean, I'm very interested in this question of stress. We have perceived stress. We've assessed food insecurity. So I did actually want to do a specific study of food insecurity and the impact on responses to food cues. We don't have lots of kind of food insecure participants in my study. So to try and get at that, I've more kind of tried to think about income. And we have also calculated area deprivation index based on zip codes, which may speak a little bit more to the kind of environmental kind of aspects of this. We do find that these measures kind of cluster together. So for the paper that I worked on with Lorette, and something we're working on now, we've kind of created a social disadvantage score, and we're able to kind of use that as a metric to see how that impacts these different food measures. I'll say a finding that when I was kind of exploring these food insecurity effects earlier on that struck me was, so I had a hypothesis that, you know, food insecurity, particularly because of this kind of cyclical nature where you kind of may have food and then you don't have food, that was kind of reminiscent of an animal model for binge eating, where you kind of have these periods of deprivation, and that might set you up for binge eating. But we actually did not find kind of evidence of eating disorder type behavior from kind of self-report in our sample, which I kind of interpreted as being that actually some of these kind of eating disorders, I mean, they may be things which are expressed in a kind of higher income setting when they're a bit more connected to kind of worry about weight, or, you know, maybe that's in a way you kind of have to have that more kind of resource-filled environment in order to sort of express that psychological type pathology. Robert probably can speak better to that than I can. So I thought, for example, in that case, there wasn't a kind of direct, you know, the prediction didn't kind of quite, the data didn't quite follow my prediction. In terms of like how to address this going forward, I mean, my you know, go-to solution is kind of a bigger sample. So certainly in this longitudinal study, it is kind of, there is diversity. Obviously, the people who are going to kind of keep coming back with their kids tend to be the kind of higher income, but they're doing a lot at that end. So this data collection is not happening at Hopkins, this is happening at Brown, and they're trying to do kind of outreach to get some lower SES samples in, and that sample is going to be about 1,200. So then I think we have much more opportunity to get into it. We could try and do some more kind of specific sampling here, but my approach is always, I get a bit concerned when I start trying to kind of hold one thing constant, let the others vary, because I think it's a bit of a whack-a-mole situation and I might get kind of imbalances elsewhere. So I've always been a bit nervous about doing that for sort of my smaller kind of case control studies. But my answer to that would be kind of a larger sample and making sure that we get variation at the kind of lower SES end, and obviously do a better job of kind of the phenotyping in terms of stress and things like that.
Thank you very much. So instead of asking to unmute everyone, why don't you click on the blue hand if you have questions from the digital floor. And then we will have the last round, a very quick highlight from the panel. Any question? I don't see any blue hand. Okay. So thank you so much, Suzanne. I think that the presentation and your work overall, which I always admire a lot, has very much this bring to society quality, and that it has been a pleasure to progressively develop a collaborative relationship with, in fact, many of us here. What I would like now, maybe if you have something to add, Suzanne, before we have a quick panel contribution.
Well, I'm just kind of thinking about stress. Spencer's got me going on stress now. I mean, I was just thinking, I mean, one of course can have variation in kind of stress, even with in the higher kind of income setting. So I think we can kind of look at both independently, but then the other question is, I think even if say with the perceived stress score, if someone is checking kind of a high number, in a kind of more resource-filled setting, then that may mean something different to somebody's kind of score in a low income setting. So that's just a kind of extra thought about that.
Okay, thank you. Spencer, you want
I think I'll just add to that. One,
exactly.
I find it really interesting, some of the work that's coming out that, disentangles chronic stress and its effects on health versus perceived stress or stress appraisal. And how important context is, and having, say, social resources, particularly for a family, in how they might interpret as chronic stress. So, you could have high levels of food insecurity, but if you have a family, a strong social support network, that level of insecurity may not really affect you because you have those social resources to draw on. So, I think, moving forward, really separating or disentangling chronic stress from stress appraisal is a, important element.
I think that's right. And we're also interested in, so I know there's a measure of, ACE, so adverse child experiences, but then there's also a measure of protective, I can't remember what it's called, but protective child experiences, which I think may get to maybe some of that probably picks up on a little bit of, being able to have support to deal with things. And I think that's really important. In general, we're kind of missing that kind of more positive side in the research so far. So your point is well taken.
Thank you.
Okay, thank you very much. So I would like to have a quick round from the panel with two quick questions. We have spent 2 hours here. First hour, very much appreciating Suzanne's work. A sentence on the key takeaway of those 2 hours. But a sentence on the key giveaway in terms of what are your insights in advancing this convergent science within the domain of obesity or otherwise. And I'm going by the order of the picture on the page I have. Daiva, and then Patricia, Anita, Cecilia, Robert, and Spencer. Daiva.
Okay, so for the first point, I think this 2-hour session has really nicely illustrated how complex and how interrelated so many concepts are, and just how food and eating is so central to many diverse issues, not just health, but also social behavioral. I find it really fascinating to think about, especially because food is an essential. And I think the time that we're in right now, the COVID pandemic, is such an interesting time to think about this issue with food. And I know a lot of us are working on that. So I really like how we've illustrated this interconnectedness through food in our 2 hours. And the second point, Laurette, you wanted to talk about how this informs our own research program. Is that correct?
Either your own or the Convergent Science program. You'll give away for how to move next step.
To move next step. So for myself, really, I'm interested in taste and sensory perception. So I would really like to study more deeply. Taste is really a fundamental. If you don't like the food, you're not going to eat it. Or if you really like it, you'll probably eat quite a bit of it. So, and what's really interesting is your sense of taste seems to be modulated by the habitual diet and what someone is used to eating. And if you change that, you do a dietary intervention, completely change their dietary intake, you can change their sense of taste. So I really plan to study this in a lot more detail, being very reflective of a lot of the topics that were discussed today. And thank you, Susan. I really appreciated this 2 hours.
Okay, Patricia.
Yes, I think that the take-home message to me is the individual differences that happen at different levels, as Suzanne showed, at the brain level, at the behavioral level. And I think this is important for us to be mindful about, and to especially in our research programs. And I think that also the integration between basic science through animal models and human research is my take on this, like for my reality, I guess. Thank you, Suzanne, and thank you, Laurette, for the session.
And your giveaway, what is the immediate next step?
I think that, as Susan said, we need samples that can integrate all these different variables. So large samples, probably with a high variability in all these features that we talked about. That's what we need, I guess.
Anita, take away, give away.
So I actually want to continue talking about what Patricia just said, because I was thinking along the same way. So I think what this panel made clear is that to really successfully realize this kind of multi-layer approach, you need to have expertise from different people. And I feel just in practical steps, what that means is that you have to have this kind of multi-center studies that brings together expertise from these different areas, because we all have a specific set of expertise, but really taking advantage of all of these levels, you need to basically bring these people together. And again, I can only echo Patricia's point with this kind of big data and big samples that we need to really address the variance at all of these levels. And in terms of implications from my research, what I find interesting also, and I think that also got clear in the panel discussions, people talk a lot about concepts such as impulsivity, self-regulation, control. And depending on the background of the people who talk about it, they mean very different things about it. And so what I also think is that we need to understand the nature, the mechanism at these kind of different levels of data, what it actually means to really develop these kind of interventions or use it for diagnostic purposes or a translational research or whatnot. And I feel like that's another big agenda point.
That's a very good giveaway. That's very good. Cecilia.
Well, I think what I take from all this is that that individual differences in the developmental trajectories can confer risk in general to behavioral abnormalities. And I was thinking also, for example, to responsiveness of cues that you mentioned, to cues associated with food. I mean, also cues associated with drugs. And maybe we can think about this convergence in terms of prevention and intervention strategies as well. You know, that one intervention can help for one, but also for another in individuals who are at risk.
As your giveaway. We shift to Robert now, and then Spencer.
Well, I guess one thing that I'm thinking about is just how important it is for people working in obesity from the sort of metabolic side, how important it is for them to really work together with people on the neuroscience side. Because as we've seen today, if you don't integrate those two basic things, which is how the medical system is currently constructed, it's still a little bit separate in terms of those two ways of coming at it. It's going to be very hard for us to make progress. So I think that's both a takeaway and a giveaway, that everyone in the obesity area needs to be strong in both areas, and we need to be talking to each other, and we need to develop a whole group of new upcoming scientists and clinicians who are well informed of both sides to that story, so that they can really make the best treatments, preventions, and just understanding of the problem.
Thank you. Spencer, and you would have the last word. It's 1 o'clock. Spencer. Spencer, are you still there? Oh, he may be
I'm still here. Yeah,
okay,
sorry.
Go ahead.
So I was just going to say, I'm just generally echoing some of the previous comments about the need for interdisciplinary teams. But I guess my takeaway is more just generally the complexity of all this. And you mentioned yourself earlier, the idea of the life course perspective, and really being able to look at this not just cross-sectionally within a family or a family and children, but across generations. And there's going to be certainly challenges with a sample, even if you have larger sample size. that really getting the variability to look at some of the issues that might come up, and particularly, again, across generations. And, I guess I'll put a plug in for some of the work that Lorette is leading around the agent-based models and how we might use more complex systems approaches to model these relationships and think, about the types of interventions that might go to, improve children's outcomes. So that's where I'll end. Plug in for you for your work on that.
Thank you, Spencer. Thank you, Suzanne. You have been wonderful. Thank you to the panel. It has been very insightful. We will be having, since many of us are at home, we will not be interrupting the series for the summer. We will be having one in July and one in August, with the presenter almost confirmed. You will hear from us soon. And a reminder that the recording of all our webinars are available on MCCHE website. Thank you very much. Thank you, Suzanne.
Everyone.
We'll shift back to you. It has been a pleasure. Thank you very, very much. And we'll let you go back to your little one. Bye-bye. Stay safe,
Thanks,
everybody.
everyone. Bye.
Thank you, Lorraine.
Thank you.
Thank you.
Bye.
Bye.